talk, so Paul Petros. I'll give you a little bit more of his background than you might expect. There are a bunch of undergraduates here. You can kind of see a little bit as you see his sort of development as a scientist, his CV or his bio go, but it's not a straight line from I want to be a scientist to right here. He started out undergrad at Waterloo in chemistry and did a bunch of co-op stuff on purification of water and petrochemicals and things like that. Sort of indirectly related to cancer. Very relevant now that we all own a bit of the trans-chemical, the trans-health. How you clean the water and what do you do about petrochemicals. But he did a work term at Michigan State University with computer models of drug response, and this really got him turned on to this idea that you can do this computational work. So his undergrad work, he did a thesis, and it won first place at the National Undergraduate Chemistry Conference, and then he started his PhD at Terra Institute for Cancer did his PhD at Terra Cancer Institute. That was such a bad name, right? Mm-hmm. I was there, and I still tripped it up. OCI. And when he finished there, he started his independent research at Terra Institute for Cancer Research, and he's there today. He co-leads the Canadian Prostate Cancer Genome Network and also leads the International Consortium optimizing algorithms for genomic data analysis. So Paul's been very successful in his publications. I've been on grant panels with Paul for prostate cancer, and always insightful. He's the Terry Fox Research Institute investigator and has been named the Prostate Cancer Canada Rising Star.
Thank you. That makes me feel like I need to go back to the lab and do more work. Can somebody at the back wave if you can hear me if I speak this loud? Awesome. So we're going to talk today about something simple, which is why aren't all cancers the same? So it's a big question. Why do we see heterogeneity in cancer? And I'm going to try to give a general solution to what that might be, why we see that variability. And to do that, I'm going to cover a couple of different topics. First, I'll give an overview of the clinical space in which I'm interested in. And then I'll start talking to you about how we predict differences in cancer aggressivity. And then I'll try to get back to its origins, where it all might start. So with that in mind, if we take a look at cancer, in any type of cancer, when the cancer is localized to its tissue, there's this huge range in survival. So what you're looking at there is a Kaplan-Meier curve. The y-axis is the fraction of people alive. The x-axis is the time since diagnosis. And so at the beginning of the study, 100% of people are alive, and that's diagnosis. And over time, people are dying of their disease. This is lung cancer, but of course you could see the same plot for any tumor type. And what's really notable about this is that right over here, you can see a group of patients who are having an early relapse of their disease. And that relapse is happening despite the best attempts to cure their disease with surgery, radiotherapy, and sometimes adjuvant chemotherapy. By contrast, there's another group of patients who have long-term, durable, good outcomes. We might even say cures. These patients look like their treatment was successful, and in some cases, maybe they were even overtreated. And that's the fundamental question that I want to ask. If we can take a look at time zero and identify a group of patients who have tumors that are potentially curable but are undertreated, and another group of patients who have tumors that are not only curable but are potentially overtreated and would be good candidates for de-intensification of therapy. That's the idea. And so we're trying to tackle tumors not when they're metastatic and have moved outside of the organ and are very difficult to treat, but when they're localized and we can maximize chances for cure. And this isn't a new idea. Of course, it's actually an old one with people trying to do this from genomic data. And initially, people have done these studies based on a relatively straightforward hypothesis, that if we looked at any solid tumor type, there are a small number of subtypes. We've heard about the subtypes of breast cancer a few minutes ago. And those subtypes will have distinctive molecular profiles, and those distinctive molecular profiles will be associated with distinctive outcomes or prognoses. And to a computational person, I've just screamed at you several times, pattern discovery or unsupervised machine learning. But you all know this because you've seen plots that look like this. This is the Seminal study in breast cancer, published September 11th, 2001 by Chuck Soterio's group at the University of North Carolina. And what they did is take old-fashioned microarray technology, stuff that you could do for $50 today, and they took a set of just 50 breast cancers. And for each of those, they measured the levels of about 8,000 genes. And as soon as you look at the data, it just immediately pops out that there are these subtypes of breast cancer that are really distinct. And those subtypes of breast cancer are not just distinct in how they look molecularly, but they're associated with massive differences in outcome. Where here, the blue luminal A patients have long-term durable good outcomes, and the red triple negative patients have poor prognosis. And in fact, it's not just that these are good and poor outcomes, but they are going to respond differently to treatments. And in different forms, these are exactly the way in which we think today about breast cancer subtypes, looking at receptor status to be able to make decisions around how patients should be treated. But that's not true for most types of cancer. At almost the exact same time, David Beer's group at the University of Michigan did an identical study in lung cancer. You can see they found 3 clusters, 3 subtypes of lung cancer, which have very different survival. The first thing that I did in my PhD was to show that these subtypes are replicable in Toronto, and of course that didn't work. Actually, happily, I guess, it turned out that they were also not replicable in New York or in Boston or in Japan, and so we started to say, well, that's weird. Why would the subtypes of lung cancer that are incident in the population of Toronto be entirely different than those in Michigan or in Boston or in New York? And these studies came to their head in a really important paper published in New England by a group at Duke University. What you're seeing here is this fascinating empirical Bayes clustering technique, which I would take a long time to describe to you, but like the most perfect survival curves that you will ever see in your entire life. They're perfect because they're fraudulent, and the study is important because after they did these studies, a series of clinical trials had been started to prognose patients and change their treatment on the basis of this biomarker. And now you can think, we have this hypothesis, and the only way we can get the hypothesis to be true is to have fraud that is so large that leads clinical trials to have to be stopped, and major problems for the research community, because now we don't know which papers could be trusted from this group. In the end, they retracted 16 separate studies, and this led the field to really consider, okay, why is our hypothesis wrong? And a plot like this really shows clearly what's going on. Each of the individual rows is a separate patient. So these are all rows. And these are just 6 genes. Carefully chosen 6 genes, but just 6 genes. And if you ask yourself, how many clusters or subtypes do you see there? The answer is definitely not 1 or 2 or 3. You might think you see 20 or 30. And today we think that there are a couple of 100 different expression subtypes of lung cancer. If you think there's a couple of 100 different expression subtypes, how are we going to see those to be reproducible? It's hard with these studies of a couple of 100 patients to be able to actually come up with good biomarker studies. And in fact, truthfully, we should have listened to our statistician colleagues for years who had been telling us, we don't understand how you think this is going to work. We think you're going to need sample sizes up in the many thousands of patients before this has any chance of working. So the field pivoted and said, we need to restate our hypothesis. Instead, we would say that for any cancer type, there are a large number of distinct subtypes. And these distinct subtypes have overlapping molecular profiles. Not distinctive, but there are still distinct outcomes. So instead of having this subtype intermediation, we should disintermediate subtypes. We don't care about it. What we really care about is predicting outcome, and therefore we should apply supervised machine learning. And this is the origin of the use of things like random forests and support vector machines in computational biology in the mid-2000s. A rise of techniques trying to take things that are used in everyday life. The techniques that we use to have our elevators run, to have search suggestions on Amazon, or if you like this video, you might like that video on YouTube, Google, traffic lights, self-driving cars. All that stuff is the same technology that's used here. And I'm going to skip like 8 years of interesting research as computational biologists learned how to do this well, and kind of get to the end point, which is we found out how to do machine learning on genomic data quite well. If you give a computational biologist a data set, we can say that we can develop a biomarker that is within the top 99.9 percentile of all possible biomarkers from that data set. Phrased differently, There's no additional information to be extracted from a data set that we can't get out. But the problem is the biomarkers that maximize that information from any one data set are imperfect. This was, until a couple of years ago, the best available biomarker for lung cancer. And it's good. That's certainly a big difference in survival between patients on the blue curve and patients on the red curve. And not only is there a big difference in survival, but it's nicely statistically significant, so you can publish it in a good journal and be happy. The problem is precision medicine is ultimately denying therapy to a subset of patients. It's telling a person in the blue group, we think you have a good outcome, therefore we do not want to give you a certain treatment. That patient is therefore going to ask their clinician, sure, so how confident are you that I'm really in the blue group and not in the red group? And with this nicely statistically significant biomarker, the clinician would say 66%, and the patient would say, no, I'm not going to listen to your biomarker. And that's a very reasonable thing to do. Biomarkers are not useful when they have this kind of accuracy. In fact, surveys suggest we need biomarkers with about 80% accuracy before they will reach broad acceptance by both patients and clinicians. And so if we can't get there from an individual data set, of course the solution is merge multiple data sets. And that requires us to think hard about how to develop good biomarkers, how to create biomarkers that are not just accurate, but can also be extensible to handle different data types in single types of assays, that will handle diverse problems because clinical care pathways are not single decisions, that incorporate known clinical features into the modeling, so we don't reinvent the wheel and pretend that all tumors are the same or that we don't know about differences in how cancer presents. And of course, it should probably link to treatment options, and in a socialized healthcare system, probably even to quality of life and cost considerations, so that we're making decisions in a balanced way across the healthcare system. And the other thing we should be doing is making sure that these are fast. One of the other areas I work on is in transplant, lung and kidney transplant, and in those areas, you have to make a decision in an hour. That's not a decision about let's run something on the cluster for a day. It's an organ comes in, and you need to do genomics and computational biology in less than 60 minutes. That immediately starts to place restrictions on how we think about actually doing the analysis. Considerations like these led the field to think, well, we need to generate data sets that are large with lots of patients and lots of data types, that are extensive multimodal data sets. And a series of projects around the world were created to do that. This is called the International Cancer Genome Consortium, of which we probably know the TCGA in the US is one of the major member projects, and it's now looked at about 75 types of cancer working in about 30 countries around the world. And you can see up there at the bottom of Canada is the Canadian Prostate Cancer Project. That's the Canadian Prostate Cancer Genome Network, CPC Gene, which I lead, and that's what I'm going to be talking to you about for the first part of the talk. You might ask why you would study prostate cancer, and the reasons are simple. The incidence is extremely high. 1 in 7 men will get it over the course of our lifetime, so in a room like this, it's 10 men, 10 of us are going to get prostate cancer. That's a large burden. And of those 10 of us, fortunately, likely only 2 are going to die of their disease. The other 8 will be cured or will die with their disease. And unlike almost any other tumor type, we're incredibly good at diagnosing prostate cancer. This plot here on the right is showing you the incidence of prostate cancer over time in the population. And you can see this striking doubling in the early 1990s. That corresponds to the introduction of PSA, a simple blood test, $20 test that is incredibly sensitive at detecting prostate cancer, just not very specific. And you can tell it's not specific because the green curve for mortality barely changes, so we double the number of tumors that are diagnosed without saving many lives. And this means that we've got very large overdiagnosis of indolent prostate tumors that are never going to kill somebody. And in fact, the management of prostate cancer is perhaps the simplest of the major tumor types. It's the only cancer type for which radiology, imaging, is not part of the standard clinical workup in localized tumors. Instead, we use PSA tests. We use a biopsy which is taken transrectally, and as a result, it has a 3% chance of sending a man to the emergency room with sepsis, bacterial infection. To put that in context, prostate cancer biopsies are on track to become the single most common medical procedure in Canada by 2030. with a 3% chance of sending a guy to the emergency room. On top of that, we use a digital rectal exam in lieu of imaging. And a digital rectal exam means a physician inserts a lubricated finger into a man's anus, palpates the prostate, and tries to determine if there's a bump to see the presence and extent of the disease. And that's it. That is current state-of-art treatment in prostate cancer, prognosis in prostate cancer. And those criteria are used to group men into low, intermediate, and high risk, and that's it. And that means that there's an opportunity to do a lot better. But if there's one thing I want you to take away from this talk, it's how inspired we should be by our clinical colleagues. Those simple, stupid criteria separate patients into groups with vastly different treatment outcomes. You can see here the low-risk patients have very, very good outcomes. So good that they're treated by active surveillance, just monitoring of the tumor for progression in a protocol developed here in Canada by Laurie Klotz's group at Sunnybrook, for which he won the Order of Canada a couple of years ago. Patients in the intermediate risk group are treated with definitive local therapy, surgery or radiotherapy at equipoise. The high-risk patients receive definitive local therapy, typically plus adjuvant hormone therapy. Okay, if you're going to start doing a large project to investigate these tumors, you might think that the right place to start is in the middle, and that's what we did. We started in the intermediate risk group of patients, where a third of patients are cured by their existing treatment, a third are overtreated, and a third are undertreated. So it's a perfect classification problem. We developed a large cohort. This is Movember's largest funding initiative ever, and it's to understand the genomics of these intermediate risk tumors and how to make better clinical risk predictions. All right, so what's the things that we actually did? Well, the first thing we did is decide not to work in isolation. We developed an international consortium of the prostate cancer genomics projects, and actually this consortium moves slowly sometimes, but has its first major publications coming out this year. And we're in fact in the middle of a harmonized analysis of 3,000 prostate cancer genomes, for which I lead the bioinformatics, and there's a number of downstream working groups. But what did we actually find out about making predictions? Well, the first thing that we did was figure out that we needed to understand what the prostate itself is like. The prostate is the size of a walnut, so we chose to look at it like it's an egg. And we cut through each layer like an egg slicer, and that's what you're seeing here. The layers of the prostate laid out. And for each of those layers, then we would go in and identify in this layer what are the different regions of tumor. And our pathologist would look at them under the microscope, under an H&E, and identify here's this little region of what's called Gleason pattern 4 cancer, and that would be either manually macro or laser capture microdissected and then subjected to all the genomics that you could want to do. We do the same for this green region and the purple regions and do this across the entire tumor. So this is a complicated plot showing that. Let's just focus on the green rectangle. The green rectangle is showing you 9 regions from one man's prostate, 9 spatially separated regions. And the bar plot on top is showing you the number of genes that are mutated in each of those regions. So here we have one region that has about 20 genes mutated and another one that has about 150. So we've got this order of magnitude variability in how many genes are mutated at the point mutation level from region to region within one man's prostate cancer. And now you can see this other region of the tumor, and you've got the same story, this large spatial variability. And of course, it's not just point mutations. It's true if we take a look at other types of mutations as well, copy number aberrations or genomic rearrangements. It doesn't matter. Anything we look at shows that spatial variability. And of course, you're looking at this and going, sure, there's variability, but does that matter from a clinical perspective? Does it going to change the way you think about biomarkers? And so this too is a complicated plot. It's showing you that man where we had 9 regions from his tumor, and each of those regions are laid out. And I'm just going to focus on this one little panel here, these cancer-related genes, and I'll blow that up. You can see this line here. That's the diagnostic biopsy, the one that was used to identify that the patient had the tumor. These other 8 are taken out surgically after the patient was treated. You can see a green rectangle here for PIK3CA. That's a non-synonymous point mutation. It's H1054, so it's an activating, druggable point mutation. This man is one of the 1% of prostate cancers that has an actionable mutation when localized. We can also see that blue dot. That's for deletion of P53, an adverse prognostic factor for radiotherapy, so we've got a clear story. You could think about putting this man on a basket trial for a targeted agent, but you definitely probably don't want to give him radiotherapy. definitely probably. You can also see that if you take a look across the other 8 regions of the prostate, my story gets a lot worse, because only one of the 8 regions has the PIK3CA mutation, so the rest of the tumor is not going to respond. The p53 deletion is only in 3 of the other 8 regions. There's a bunch of regions which have a deletion in BRCA1, which is just bad. If we superimpose that on pathologic aggressivity, it turns out that the BRCA1 deleted regions are the more aggressive looking under the microscope, and unfortunately this man has had a relapse of his disease, and no points to anybody for guessing which region gave rise to the relapse. But it's that guy, which had none of the mutations that we thought were actionable. So that's a problem, because how are you supposed to make a decision about treating a guy from a biopsy where you don't want to make the decision after you cut out the whole prostate that he didn't need treatment, when the biopsy is not representative of the entire tumor? And it gets a little worse. Here's a man where we have 4 regions from his prostate. You can see at the top these 4 regions, and this is the genome laid out from chromosomes 1 to Y. And these blue dashes are deletions, regions of the genome that have been deleted. And you can see 2 regions have deletions on chromosome 8 and 16, 2 on chromosome 19. And interestingly, they don't share anything in common. You'll also notice if we take a look at the genes that are mutated in these regions, they also don't share anything in common. And what's happened here is that this man doesn't have prostate cancer. The man has 2 genetically distinct tumors within his prostate. And that makes a lot of sense. 1 in 7 men get prostate cancer in their lifetime, so 1 in 49 get 2. And I didn't account for genetic predisposition, environmental effects, lifestyle, selection bias, etc. So now we have this problem where which of those tumors do we treat? How do we even find that the man has 2 tumors if we don't have good imaging? How do we handle this problem when you can't make good predictions about outcome from a biopsy, but you need to make the predictions before you do the surgery? This is the first result we got in a 7-year, $20 million project. It is deeply depressing because it basically says a lot of what you want to do is not going to work. So Rob Bristow, the clinician scientist who's co-leading it with me at the time, and I were depressed and having a beer. And we were talking about what do we need to do to fix this? And literally, I was like, we're not smart enough to solve this. And Rob says, oh my gosh, you're brilliant. We need to hire a really smart graduate student. I'm not joking. And so we did. Emily Lalonde looked at these data, and we didn't tell her that we were thought we were screwed. And so she said, I know exactly how to solve this problem. This is what you wanted. She took the data from pre-treatment biopsies, and she said, first thing I'm going to do is I'm going to focus on copy number profiles, because those show the least spatial variability. Still lots, but the least. And I'm going to identify common trends in that data set. And you can see here, there's a couple of 100 patients laid out, and they sort of fit into patterns. And I'll make those patterns more clear. You can see these patients in the cluster 4 have very few mutations, these guys over here. There's another group of patients that have a gain on chromosome 7 and a couple of different patterns there. And what's interesting about those patterns is they're directly associated with outcome, with one group having good outcomes, 2 groups having rapid bad outcomes, and 1 group having long-term bad outcomes. And the interesting thing here is this red group of patients. That red group of patients, cluster 4 with few mutations. So a prostate cancer with few mutations has good outcome. That sounds obvious, and I know you're thinking that sounds obvious because that's what I told Emily was, that sounds obvious. That must be known. So she went ahead and looked in the literature and didn't find anything. And I said, well, that's still obvious, so this must be a general thing for every tumor type. So I made her look at 37 tumor types, and it turns out that this is true for 2 tumor types, breast and prostate. It goes the opposite direction in ovarian cancer, and it's not clear why it shows no or very trend effects in every other cancer type. So you can come up with the simplest, stupidest biomarker in the world, called it PGA to give it a nice name, but you just count the number of mutations. You just take a look at the genome, count the number of mutations, divide patients into 2 groups, and that's 70% accurate at identifying patients who are going to have a relapse in their prostate cancer. But Emily wasn't done. She said, well, I know the next thing you're probably thinking about is that you need to take a look at other trends that are consistent across the prostate. So what I'm going to look at is the levels of oxygen, because oxygen is involved in response to radiation. Radiation is a common way of treating prostate cancer, so this will be simple. And she immediately did the correlation of genomic instability with the levels of oxygen, here measured using needle electrodes into the prostate, and you can see that there is absolutely no correlation. And while Rob and I go off and continue to be depressed, she says, don't be depressed. This isn't what you need to be looking at. Instead, what you need to be looking at is how this works together with genomic instability. And when she did that, something basically magical happens. You can see here, these are the patients that have normal levels of oxygen in their tumor and relatively stable genomes. They have very good outcomes, and synergistically pulling apart with very poor outcomes are those patients that have low levels of oxygen and highly unstable genomes. These are both required processes. It's not just about the genome, it's also about the cellular microenvironment. That now says, well, okay, I can probably come up with a biomarker to do this, but you probably don't want to stick a needle into each man's prostate to do an electrode oxygen measurement, so you might need to find a better way of measuring molecular oxygen. We did that exploiting TCGA pan-cancer data and looked at the levels of hypoxia across a series of tumor types. You can see here thyroid and prostate cancer are two of the most normoxic tumor types of all, whereas here head and neck squamous cell and lung squamous cell tumors are two of the most hypoxic. We could see in almost every tumor type that we looked at clear profiles of individual mutations that were correlated with hypoxia. So this is kidney renal clear cell, tumors laid out from the most to the least hypoxic, and you can see each of these mutations is biased towards more hypoxic tumors. These are breast cancers, and you can see P53, for example, tightly associated with tumor hypoxia. So Emily was able to use that type of information to come up with a molecular predictor of hypoxia, merge that with PGA, and come up with a biomarker that's almost 80% accurate. Somebody who's paying close attention will notice that the area under the curve, which is maybe a better metric of accuracy, is a little worse. And our reviewers unfortunately noticed that and said, we want you to validate this in a larger cohort. We don't believe your biomarker is as good as you say it is. So working with a company in Vancouver, GenomeDx, we validated this in 1,000 patients. And happily, in our validation cohort, it does even better with an AUC of just at 0.8, right at that clinical threshold. All of the other black lines are commercially available or academic biomarkers, and current clinical practice is right over there at 0.6. So you can see, for these patients, we've improved 20% of intermediate risk prostate cancer patients' treatment decision making. In North America, that's about 130,000 men a year, so large, large numbers. We then went and reduced this to a $200 nanostring assay and validated in a series of other large cohorts. And you can see in each of those, it's got this AUC of about 0.8, so about 80% accurate. Great. 80% isn't 100%, obviously. And so what are we missing? What we're missing is 20%. And so what is that 20% coming from? I naturally figured that the rest of the 20% was coming from all the stuff we didn't look at. We had looked at copy number aberrations for this 80%, so the rest of the genome explained the other 20%. So we went off and did a large-scale study of that. We spent about $5 million sequencing 200 prostate tumors, doing their methylomes and transcriptomes to try to understand what the origins of the rest of the 20% was. We found like a series of absolutely fascinating things. Just one example of it, here are prostate tumors laid out. This is the number of mutations, and you can see these tumors here are a prostate cancer which in the 3 billion bases of its genome has about 50 mutations. It's hard to understand how 50 changes make something into a cancer in the first place, and there's no other adult tumor that has this low a mutation rate, just a couple of pediatric tumors actually. So that's fascinating, and there's a number of other things. Maybe most importantly, we identified a set of 40 different events that drive aggressive prostate cancer. These range from individual mutations to processes like chromosomal shattering, chromothripsis. We put those together into a model with a biomarker and some pretty good machine learning to be able to predict patient outcome. And at the end of it, we made a biomarker which was 83% accurate. So what we successfully did was turned our $200 80% accurate assay into a $9,000 80% accurate assay. So not so good. If anybody ever says to you, can't publish negative results, that's not true. So that's great. The other 80%, the other 20% is clearly not in the rest of the nuclear genome. I'm going to tell you about some of the things that are not explaining that, and I'm going to go through this part a little quickly. The next thing that we thought is that we had to look at the second genome of the human cell, the mitochondria, so we went ahead and sequenced the mitochondria of 300 patients. You can identify mitochondrial hotspots, mutational hotspots in those tumors. We found that those mutational hotspots correlate to what's happening in the nuclear genome. Those are the nuclear genome driver events. These are genes in the mitochondria. Each time you see a red dot with a black background, that's co-occurrence of a nuclear and a mitochondrial phenotype, showing that you can't understand what's happening in subtypes of cancer without considering the nucleus and the mitochondria together. And these blue dots are mutually co-exclusive mutations, where they actually seem to be only one is needed to drive a certain phenotype. These are associated with outcome in a synergistic way. For example, patients with both a mitochondrial origin of replication and MYC mutation have very aggressive mutational clinical characteristics. Those with one are sort of intermediate, and those with neither are better. This explains none of the missing outcomes, so we then said we need to study a different analyte. We took a look at the methylome landscape of 620 tumors. We looked at H3K27 acetyl of a series of 40 of these tumors. We put those together into a biomarker that was 75% accurate. So we then said we needed to sequence the transcriptomes. We sequenced the transcriptome of 155 tumors to the highest depth that anybody has ever sequenced. Incidentally, we only got an accuracy of 70% from the transcriptome, so we're going in the wrong direction. We found out that there's widespread RNA circularization, so that many of these RNAs, actually hundreds of different transcripts per cell, are circularizing through backsplicing events. And so you have these circular RNAs that can actually make peptides or serve as microRNA sinks. We went ahead and did a series of functional validation on this using shRNA screens and showed that many of these are in fact essential to cell proliferation, and so we have circularized transcripts in our cells that are essential. Anyway, doesn't explain any of it, so we needed to look at something that's more functional than RNA. So we took a series of 55 prostate tumors, did whole proteome mass spec on each of those. We found out that there's pretty good correlation with the RNA, but pretty good correlation is not that great. So the R is about 0.7, so only about 70% correlation. And actually, if you take a look at the values on this axis, they're not well related. So Ephrin B2 is an example of a gene that's changed about 500-fold at the protein level, but only 2-fold at the RNA level. So the differences in magnitude of protein changes and RNA changes are huge. But anyway, you can show that correlation depends on the abundance of a protein, put that together into biomarkers and identify the best proteomics biomarkers, and they get right into the 78% accurate. So that doesn't help us at all. So a lot of things that failed. So in the end, last year, we finally figured out what we should be looking at. We figured out that if you take a look at a biopsy is ultimately a snapshot in time in the life history of a tumor. And the tumor might have been developing for a decade before that. And what doesn't just matter is, in a nicely quantum mechanical way, it's not just where you are today, but it's where you're going. And if you think about that, then you need to be able to estimate tumor evolution. How do you estimate tumor evolution when you only have a single snapshot in time? We can't tell patients we're not going to treat you if you have what looks like an aggressive tumor. So we need to do computational inference in some way. And the best way to do that is to take a look at the bulk set of the total number of cells and estimate how many different populations are present in that tumor. What is the subclonal diversity? That had not been done before, so we set up an international crowdsourced challenge to be able to make those estimates, working with groups in Oxford, the Crick, the Sanger, UC Santa Cruz, Oregon Health Sciences. We were able to run a collaborative challenge across the world to identify methods that would solve this problem. We then went ahead and applied it to our prostate tumors, and now we could construct a phylogenetic tree for every one of our tumors. So here we've got a couple of hundred tumors, and for each of them we've got a phylogenetic tree. This is the normal. The branch length is telling you the number of mutations, and these guys are monoclonal, just one type of population within the tumor. These tumors are polyclonal. You can see 2, 3, 4, 2, 3, 1, 2, different populations within the tumor. You'll also notice that most of the tumors, these are the numbers, are linear, and only a small subset are branched, and we'll come back to that. And so you can come up with this landscape of how different prostate tumors are evolving. That allows us to put individual mutations into the evolutionary context of when they occur, and we can start to ask how mutational processes change over evolutionary time. So you can see here, for example, there is this gold trend, which is dropping over evolutionary time. That's the mutational process of human aging, a deamination process that's naturally occurring in cells, and it becomes less important as tumors evolve. By contrast, you can see this red process that's increasing over evolutionary time. That's the BRCAness profile, homologous recombination defects. Becomes more important as tumors evolve. So if we just take that information and put it in the context of patient survival, something amazing happens. That gold curve at the very top, those are the patients that have a monoclonal index biopsy. There's just one cellular population. Those patients basically never have a relapse of their tumor. It's a 4% rate. By contrast, if we take the patients that have subclonal diversity and we divide them by that $200 hypoxia genomic instability marker, we can see two groups come out. This blue group, which has about a 20% chance of relapse, and this red group with almost a 70% chance of relapse. This is clear. These patients look like they should be on active surveillance. They look like low-risk patients, and they probably shouldn't be treated. These patients look like they're being cured by existing therapy, given the efficacy of radiotherapy, the fact that we don't know whether a patient would have a radio-resistant tumor and positive margins at surgery. And these patients look like high-risk patients. They should be getting adjuvant therapy up front. But at time zero, all of these patients were diagnosed as intermediate-risk prostate cancer. So we can make those predictions by merging three things: the microenvironment, the genomic features, and tumor evolution. Okay, but I promised to tell you how we're getting there. And so I'll do that in the last couple of minutes quickly and talk about why some tumors are aggressive. When we saw these data, the first thing that I thought of was, if it's starting all the way back with tumor evolution, then it must be a feature of something that predates tumor evolution. So I hypothesized that this was a function of the germline, that something in our germline was triggering different evolutionary paths. It's a hypothesis. To test that hypothesis, I said, let me find the largest germline difference that I could, which is sex differences. Entire chromosome different, and chromosome X is pretty darn large, so it's like 200 million bases that differ between males and females. 270, I guess, if you add Y. And so that's not a new idea. Of course, looking at sex differences in cancer is long-standing. The first papers on it are like 80 years old now, and if we take at different tumor types, things like larynx or hypopharynx tumors are 5 times more common in men, whereas tumors like thyroid cancer are 3 times more common in women. So you could take a look at individual cancer types, like here lung cancer, and find individual mutation differences that are reported in these bespoke studies by individual groups looking at small cohorts. To look at this systematically, we took a series of 35 tumor types from the ICGC and TCGA projects that I talked about at the beginning. And I'm just showing you that favorite metric, PGA, genomic instability, and I'm showing you 5 tumor types, 5 randomly selected tumor types. These are not like the best or the worst or anything like that. This is AML, kidney renal clear cell, head and neck, stomach and esophagus, and sarcomas. And you can see in sarcomas, even after controlling for everything else we know about the patients, female tumors have a higher mutational burden than male tumors. In the other tumor types, it's different, with a 3-fold increase in mutational burden in AML for male-derived tumors than female-derived tumors. Okay, so differences in genomic instability, but do these influence individual genes? This is a complicated plot. What I want to highlight is this plot at the top. Each of these dashes is indicating a region of the genome that is specifically sex-biased. So you can see that there are sex biases in almost a quarter of the genome in 1 or more tumor types. And you might wonder how big these can be in effect. Are we talking about little changes? Let's focus on this panel, which is liver cancer. In liver cancer, beta-catenin is a frequent mutation, and in male-derived tumors, beta-catenin is mutated in about 40% of cases up here. In female tumors, it's mutated in only 15% of cases, so there is a 25% absolute value difference in the beta-catenin mutation rate. BAP1 is mutated in almost 20% of female tumors and only 5% of male tumors. So we're seeing huge differences in individual mutations. But of course, I was talking about biomarkers before, and you might wonder if different genes are engaged in predicting outcome. And so what you're seeing here is lung cancer, our favorite tumor type from the initial studies in patient prognosis. These are adenocarcinomas of the lung, and these are squamous cell carcinomas of the lung. Each point is an individual gene, And this is the association of that gene with outcome in men and in women. And if that looks like a random scatter plot, it's because it is. There is almost no association in the genes that are associated with outcome in men and in women in lung cancer. And that, of course, means that a lot of our biomarkers, like the first one I showed you today, are sex biased. That biomarker does quite well in tumors arising in a man and has almost no predictive accuracy in tumors arising in a woman. Sex biases are very large molecularly and therefore need to be considered in biomarker development studies. Okay, but that's a whole chromosome, that's a large thing. Can we find something smaller to look at? To do that, we focused on a developmental tumor type that's perhaps the most important tumor type for our understanding of cancer, which is retinoblastoma. Retinoblastoma was the tumor type that gave us, amongst other things, the first tumor suppressor, the unification of somatic and familial genetics, the first cooperating oncogenes, the discovery of E2Fs and cell cycle progression. I can keep going. There's a lot of things that came out of the study of retinoblastoma, and 40% of them are initiated by germline mutations in RB1, the first tumor suppressor. And that means that these tumors will have what's called a two-hit model. Their time of progression will look like they required independent hits in both tumor suppressors. To try to identify just how frequently this occurred, we sequenced 1,000 retinoblastomas working with Brenda Galley, one of the pioneers of RB research in Toronto. We found 16 tumors which did not have an RB1 mutation. And I'll skip through a lot of cool things to point out that these tumors instead had amplification of MYC-N and follow a one-hit model. They look like only one mutation was required to initiate a tumor. And now we've got a case where we've got tumors that look morphologically identical, but some of them are initiated by loss of a tumor suppressor, and some of them are initiated by gain of an oncogene. We sequenced those. We're able to show that individual mutational processes, like the BRCA process or a process associated with ApoBec-mediated hypermutation, were different between tumors with and without an RB1 mutation. We could show that their structural variant profiles look completely different. I mean, that's about as different as you can imagine. The RB1 tumors just have these aberrations around MYC-N, and by contrast, there's a wide amount of genomic instability in the RB1 deleted tumors. And when we look at the evolutionary profiles of these, they're incredibly different. Here are the RB1 wild-type MYC-N amplified tumors. Notice that both of these are monoclonal. Here are the RB1 mutant tumors. You can see not only are they polyclonal, but they're all branched. So there's this huge difference in the evolutionary complexity of these tumors. Okay, fine. RB1 is an early pediatric tumor. Can we do this in an adult late onset tumor? So of course we looked at prostate. The first thing that we looked at were prostate tumors driven by germline mutations in BRCA2. It's about 4% of prostate tumors. I'm only going to show you two quick results from this. Here, MYC is gained in 75% of germline BRCA2 prostate cancer, but only 25% of sporadic. So it's completely changing one of the most important oncogenes in prostate cancer and in cancer in general. And if you take a look at the evolutionary trees of a set of BRCA2 germline prostate cancers, there's only one thing you need to notice there. Every single one of those is branched. It was only 20 of the 293 that I showed you earlier, but it's 100% penetrant in the BRCA2 germline tumors. Completely different evolutionary characteristics. Okay, rarer variants, is this just BRCA2 or can other things do it? The first rare variant that we looked at is KLK6, a gene downstream of PSA in the same family. And this is again a complicated plot, and I'm going to just focus you on one part of this. This line here is P10, and these patients up here are divided by whether or not they have a germline SNP in KLK6. These patients do, these patients do. P10 mutations are more frequent, p-value, more frequent in patients without a germline SNP in KLK6. This SNP is phenocopying deletion of P10. That's weird. You wouldn't expect a germline SNP in a gene on another chromosome to be phenocopying deletion of an important tumor suppressor. But completely concordant with that, that germline SNP is associated with outcome. It has an odds ratio of 3.5 in large screening cohorts. It's associated with aggressive disease. And you could measure this thing for like $5 using Sanger sequencing. Like it's an easy clinical assay. That gives you a lot of hope. And actually there's a lot of bespoke reports from this. My collaborator Hinz and he at the University of Toronto and I identified another one in a long non-coding RNA, pCat19, at the same time as we found the KLK6 SNP. So to look at this deeply, Katie Houlihan, a really brilliant graduate student in my lab, said, why don't we take a look at all of the driver events we know in prostate cancer and see if there are germline SNPs that predict them. Can we find germline SNPs that predict how a tumor is going to evolve? And so this is an example of one driver event. We're back to P10. All of the SNPs that are above this line, genome laid out from chromosome 1 to Y, are individually predicting the presence of somatic P10. And so KLK6 is that guy right over there, and you can see a series of others. And you might say, okay, maybe P10 is special, so these are TMPRSS2 ERG fusions, the ETS rearrangements that happen in about half of prostate cancer, and you can see a whole bunch of those. And in fact, we estimate that we found 199 of these, and we estimate that there are at least 20,000 germline SNPs that are predicting the way that tumors will evolve. To give you an example of just how strong these can be, TSERG1L is an elongation factor, a transcriptional elongation factor that's an important driver associated with aggressive prostate cancer. If you've ever done GWAS studies, those are amazing hits. You don't get hits like that very often. It's a p-value of 10 to the minus 18 at the top. This mutation is not only, the SNP is not only associated with changes in TSERG1L, but also with allele-specific differences in its RNA and protein abundance. Those are tumor-specific, occurring only in the tumor and not in adjacent normal. And that makes you think that you might be able to predict the levels of TSERG1L from germline data alone. And so what I'm showing you here are attempts to use germline SNP data to predict the tumor status of an important oncogene in prostate cancer. And you can see, like, those aren't great predictions, right? Like, that's okay, there's clearly a correlation. But the amazing thing is, these germline SNPs were present 6 decades before these men were diagnosed with prostate cancer. So we're using information to predict what is going to happen in a tumor that is not going to evolve for 60 years. And of course, TSERG1L isn't the only story for which we can do that. We're able to do that for all of our different oncogenes and tumor suppressors that drive in prostate cancer. And do I think that we actually have 100% prediction of SPOP? No. I'm sure it's a little overfit, and the actual numbers are in the 60 to 70% range. But it speaks to a powerful influence of our germline on tumor evolution, one that may not be yet well enough understood, and one that calls for clever use of both large patient cohorts and animal model systems to understand the ways in which the host microenvironment is influencing tumor development. So what did I tell you about over the last 45 minutes? I hope I showed you that we can find ways to overcome spatial heterogeneity in cancer, that what we need to do is think about processes of different types, merging the genomic with the microenvironmental with the evolution, and that those things synergize together in really unexpected ways. I also hope that I showed you that the germline is playing a powerful role in the way that tumors evolve and is probably significantly understudied. And I know that there are similar results in other tumor types that are now in the publication process, and this likely will be a pan-cancer trend. And I think maybe from a biomarker perspective, more than anything, these data call for us not to think about treating men with one magic biomarker that's going to solve all our problems, but instead to say what we really want is like a cheap urine assay, something that has lots and lots of false positives, but that people can run at home for a couple of dollars a day. And that in the context of your germline risk, that is longitudinally updating a man's risk score to the point that it says, hey, you better go see your doctor. And then that doctor will say, yeah, you know what? We'll give you a blood test, test PSA and other circulating markers. And those circulating markers, using the prior information from the longitudinal data from the urine tests and the germline data, will now say, you know, you actually need to go for a biopsy. And that targeted biopsy again then goes through this somatic merged hypoxia genomic instability evolution marker. And so we stage things where the early assays are cheap, non-invasive, and have lots of false positives. And the later assays are expensive, invasive, and have almost no false positives. All right, so clearly I did like none of this work, and this is a large team effort. This was Movember's largest funding initiative, so I have to thank Movember, Prostate Cancer Canada, OICR, and pretty much every other Canadian funder contributed to this work. These are the people that were leading the study. Rob was the clinician scientist who was driving this with me until he moved to Manchester last year. Theo van der Kwast is the study pathologist. Tom led the proteomics component of this. Hansen led the RNA-seq component of this. And I showed you about 15 or 16 papers. So these are just the first authors of the papers that I showed you. That's my team, and thank you. I'd be happy to take questions.
That was an amazing talk, thank you.
Thank you.
So it's interesting, when you're looking at these germline SNPs that give rise to bad tumors, do they tell you anything, though, about incidence and prevalence?
Yeah. So the question was, do any of the germline SNPs which are associated with somatic phenotypes tell you about incidence and prevalence of the disease? So we did that, we looked at that question backwards. We asked if the SNPs that were associated with incidence and prevalence were associated with specific somatic phenotypes. And the answer is yes, but weakly. It appears that there are different SNPs that are associated with do you get prostate cancer or not than the ones that tell you what kinds of prostate cancer that you get. I'll give a quick hypothesis, no data behind this, for why it might be. I think that tumor genesis is such a large, complex process with so many phenotypes of immune system and evolution and that it makes sense that the SNPs that are associated with that would tend to have small effect sizes and would not necessarily be proximal to a specific molecular event in the same way that we're seeing here, where an individual somatic mutation is related to an individual genomic feature. In my head, that proximity of two molecular events relative to a molecular and a physiological one, pathological one, makes sense, but I got no data to support that.
So do you think that in totality you'd be able to put something together and predict early on that once the numbers get large enough, those will be more aggressive?
That is exactly it. So we just got funding to take a look at the Ontario Health Study, where 300,000 people from Ontario have given blood and linkage to clinical records a decade ago. And then we follow up those individuals annually over a decade. We, the OHS team, does. And now we're going to take the 4,000 men within that study who have since developed prostate cancer. We have pretreatment blood, germline genotyping, and long-term clinical trajectories. And that's exactly what we're trying to do with it now. So yeah, good question. Yeah, go for it.
That's very interesting. So when has there been any research yet or any data about people having seen germline SNPs and then the outcome one developed the prostate cancer and the other one didn't? What the environmentally or evolutionary to this event?
Yeah, so the question was, are there data where we might be able to control germline SNPs and then take a look at phenotypes, both the type of tumor that might be initiated and whether or not a tumor actually occurs?
Yeah, and how do people live in environment?
Yeah, and relate that back to microenvironmental and macroenvironmental factors. So the answer is largely no. There's not much in the way of great molecular twin studies, nor are there great environmental characteristic studies in prostate cancer. What we know is that African ancestry is a risk factor, unclear why, but plays with the germline SNP data. That obesity is a very strong risk factor. High-fat diet, which is correlated with obesity, and it's not even clear if those are separate risk factors or related, and age. So not deeply helpful. In the OHS patients, we have pretty good environmental data, and my colleague Phil Awadallah had a nice paper in Nature Communications this year where he took 2,000 patients from that cohort, did RNA sequencing on the blood, and then related it to where people lived, ethnicity, germline SNPs, and other things like that, and was able to show that a couple of atmospheric characteristics, so there's an Environment Canada atmospheric monitoring station in each postal code, and so you can take a look at things like ozone levels. And that study showed that, and I think it was ozone, was the best predictor of blood RNA levels rather than your underlying ethnicity or other characteristics that we had like BMI or things like that. So that just came out in Nature Comms a couple of months ago. So I think that's something we'll be able to look at in the OHS cohort, but I'm not even convinced 4,000 patients will be enough to pull out those effects either. But it's a great question. Yeah.
So how much noise do you think might be contributed by the immune system? So by that I mean a lot of the stuff that you're showing correlations with and great predictive value of things like mutational load.
Yeah.
Which the immune system, that's going to allow the immune system to differentiate cancers from normal tissues. And prostate cancers are a particularly interesting example where you get a lot of tissue, the prostate infiltrating lymphocytes. But there's another layer, especially there, that's the class of cancer where you'll have some patients who have good infiltration into tumors, others where you have a lot of these tumor or tissue infiltrating lymphocytes that are in the peritumoral
Yep.
area and don't actually penetrate. And so we're doing a lot of these analyses, sequencing and proteomics and stuff like that, especially when you're looking at intermediate population. You can imagine a scenario where you may have some mutations that are running the immune system, but that's also what that's doing then is increasing the number of contaminated normal cells, right?
Yeah.
That are influencing your analysis, and separately, right? Immunologists are showing that looking at the effector cells that are infiltrating these tumors also have great predictive value. So I wonder if you consider trying to combine the two.
Yep. So the question, I'll shorten that, but paraphrase quickly. The question was, what is the influence of immune cells on the way that we think about our biomarkers, and how do they introduce noise? With a couple of aspects. One is that they create contamination of normal cells, and two, immune cells are well known to harbor signal that is associated with response to treatment and the way that the tumor is managing to evade immune surveillance. And that's particularly relevant in the context of mutational burden, where the mutational burden might increase the number of neoepitopes and immune activation. So that's broadly the question. So there's a couple of pieces there. The first is that's exactly one of the reasons why we did macro, or in many cases microdissection, to make sure that we had relatively pure populations. And we can read from the sequencing data that we have 80% or more tumor cellularity and enough reads to be sure that we don't have a false negative rate associated with the normal cell infiltration. So that's just the technical artifact side. On the other side, the next question is the mutational burden changing immune cell activation? That was the first thing that we said, and we put that in as a hypothesis in the nuclear genome paper, and our reviewers said, prove it. And so we did actually quantify the number of infiltrating immune cells in each of our tumors and look for a correlation with mutation burden, and there was none. And that led us to go back and do what we should have done in the first place, which was to estimate the number of neoepitopes and how much it differed. So how much these mutations were changing what the exterior of the prostate cell might look like. And the answer is just not enough. There's not much change. And the reason is prostate cancer is what's called a C-class tumor. It's driven by double-stranded breaks. Most of the changes are changes in copy number, and there are actually not that many point mutations. The average prostate tumor has 20 genes changed at the point mutation level, and that means it's only got 1 or 2 neoepitopes. And that's just not enough, even in the differences in mutational burden, to even remotely suggest immune cell activation in localized prostate cancer. Metastatic is a different answer, and there's a series of papers coming out from the Stand Up to Cancer group in Cell in the next month or two showing that it's very, very different. But in localized tumor, that probably explains why trials have all been negative on checkpoint inhibition and activation assessment in localized disease. The last question is, do we think that there are differences in immune cell composition from prostate tumor to prostate tumor? The answer is clearly yes. We can see that both through IHC and more succinctly through the RNA-seq data, where we deconvolve those profiles. And there are clear associations of somatic phenotypes with immune cell profiles of those tumors, but we haven't done enough analysis for me to speak confidently about what we're seeing there yet, but that's definitely active work. Yeah, at the back.
Very important, but also very, very booming for a veterinary researcher, and I say that because we like to spin our research as potential models. So if we do something in the dog, we spin it as, yes, it's a great model for human medicine, right? And I think as your talk shows, the truth is that A, biology is very complex, and B, it takes a lot of money to get it at the base of that, right? So I think in a way we're kidding ourselves if we really seriously think that, and this is a bit heresy now, but that we can really like actively contribute to the human situation in a lot of ways. And so for me as an early career researcher, if you had a fraction of the money that you have, what would you advise me in terms of how can I make a meaningful contribution for a species, an animal, but then maybe also for humans?
Yeah, so it's a wonderful question. So I'll paraphrase that. It's saying what I showed you is expensive, and it relied on the value of well-annotated clinical cohorts, And how does somebody working with less funding in a veterinary or any other setting think about replicating and making sure that their work has a contribution in the way we think about healthcare? And I think the answers from my perspective are threefold. One, my lab didn't start off with a lot of money. This happened a couple of years into our lifespan. That sounds terrible. Into our existence. Oh, geez. Evolution. And I appreciate the many rejected grants that I had on the way to getting this stuff. Part one is to make sure that you pick problems that other people couldn't tackle. And I powerfully believe, that model systems are incredibly important in prostate cancer research. We don't have good xenografts, we don't have good organoids, we have terrible cell lines. Actually, like the most commonly used prostate cancer cell line is LNCAP, LN for lymph node, so it's a metastasis from a lymph node. It's like localized prostate cancer has no useful model systems. Dog, unfortunately, actually doesn't get a lot of prostate cancers, and most of them appear to be other things that have moved to the prostate, but there are a couple of breeds that do. But other animals do get prostate cancers, and I think that that would be deeply valuable. And the looking at the germline genetics says a whole lot about how we might be thinking about model systems, especially outbred ones, giving opportunities to take a look at that. And if we had a tumor type that was prevalent in dog, for example, so lymphomas would be a good example, collecting large cohorts of that is incredibly valuable in starting to understand that linkage, especially because when we take breeds of dogs, they show beautiful spatial, evolutionary, germline differences that are ideal for working out these relationships. So finding the uniqueness of your system and selling it hard really matters. And I'll give an example, and this is opinion. One of the most beautiful exemplars of that are my colleagues who study pediatric brain cancers, where they are exceedingly effective in Canada at making the argument that these are ideal model systems for understanding the aggressivity of cancer, that they are a potentially soluble clinical problem. And they do better at grant funding than I think any other tumor group in Canada. And it's not because they have as many cases or anything like that as we see in prostate or lung, especially lung, but they're able to really tightly make the argument of their unique selling point, and it resonates. So that would be one answer. Second answer is, to do studies like this requires long-term collection of unique tissue resources. It's easier in prostate because there are so many incident cases, but what I didn't tell you there is that these tumors had 10-year median follow-up at this point, and that meant that it wasn't me thinking, oh, these are the right things to do. It was brilliant clinician scientists thinking, we need to start banking these like before I was finished my PhD. And that is an opportunity to start banking tissues from different situations and wait for the genomics to get cheap enough. Wait for technology to get to the point that you can look at that large cohort. And there's lots of interest to being the first people to sequence a tumor type, but actually, we're probably at the point where you can do a good prostate cancer genome for $1,000. If somebody miraculously had 1,000 dog prostate tumors, that's a unique selling point, and it's $1 million. That's a lot, but it's not impossibly a lot. And that price in a couple of years will be $500,000. And a couple of years after that, it'll be $150,000. And suddenly this all gets a lot easier. So banking resources that you're not going to be able to analyze today, but that you can analyze in the future, I think is of deep value. The last comment, and this is still all philosophy, is I was thinking this on the way driving down, and I was wondering if somebody would ask a question like that. And I think that it's extremely valuable for a place like Guelph that has a strong school that focuses on models to identify methods to partner with academic centers that are academic medical centers that are looking at large patient volumes and see the interplay of it. And I was thinking about why that hasn't necessarily happened and what it would take for a strong vet school to partner with a hospital that had a good research program in, I'm making up places, but London, What would that take, and what are the competitive differences, and how would you form that around individual tumor types? And I think rather than treating it as these are different approaches, I think they're complementary. And you could imagine a strong veterinary aim in a Terry Fox Research Institute program project grant would be very well appreciated. And tagging along with a larger project grant that was looking at primary patients and innovative model systems and clinical studies altogether, I think would be an avenue to make sure that model system research gets better integrated. That's opinion. Maybe it's useful.